Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over a derivative of cholesterol, that is bile acid, and how this bile acid is synthesized and how the bile acid is regulated. Let's get into the details. Now before I get into bile acid synthesis, let me explain you what are the major fates of cholesterol which is synthesized in the liver. Because one of the fate of cholesterol that is made in the liver is bile acid synthesis. In that connection, we need to know what can be the other fates of cholesterol in the liver. So the bulk of cholesterol that is synthesized in the liver, it can go into one of the three fate which is given down here. One is cholesterol ester formation, other is biliary cholesterol formation, and the third is bile acid synthesis. Now, in the formation of cholesterol ester, a fatty acid will be attached to a third carbon of cholesterol to make it as a cholesterol ester, and the cholesterol ester is a major storage form of cholesterol. And the enzyme that catalyzes the esterification of cholesterol with the fatty acid is called as 𝐒𝐵𝐴 cholesterol acyl transferase. So this ACAT enzyme is going to convert cholesterol into cholesterol ester in the endoplasmic reticulum, and then that cholesterol ester will be transferred to ApoB48, which is present in the rough endoplasmic reticulum. So a lipid present, cholesterol ester which is made in the smooth endoplasmic reticulum will be transferred to a apolipoprotein B48 which is made in the rough endoplasmic reticulum and to make VLDL, very low density lipoprotein. And note that it is not just the cholesterol ester that is loaded onto ApoB48, it is also triacylglycerol which will be loaded onto ApoB48 in a similar mechanism coming from the endoplasmic reticulum. The phospholipid also loaded onto ApoB48, and vitamin E is also loaded onto ApoB48. So all that will make a VLDL, and VLDL will be secreted into the blood, and that's how a liver is going to send cholesterol ester out into the circulation in the form of VLDL. Now in the lipoprotein metabolism, I have explained you what is the fate of VLDL in the circulation. So in brief, VLDL will be metabolized into IDL, IDL will be metabolized into LDL, and LDL will taken up by the hepatic tissue and extra hepatic tissue. So that's how a cholesterol ester made in the hepatocyte is sent out of the liver to other tissues. Now, some of the free cholesterol in the liver is entering into the intestine through biliary tract. So that means whenever hepatocyte has relatively higher levels of cholesterol, free cholesterol, so it will be simply secreted into biliary canaliculi and allow that to get into the intestine, and in the intestine 55% of the secreted cholesterol will be reabsorbed and 45% will just go out of the intestine into the feces. So that means a cholesterol which is excess in the liver will be simply secreted into the bile, and the biliary cholesterol, it is metabolized in the lumen of the intestine by the bacteria into captosterol, and this is going to be present in the feces, and that's how the cholesterol is excreted out of the intestine. So these are some of the fates of cholesterol here. One is cholesterol ester, biliary cholesterol, that I have explained you now. So esterification of cholesterol in the endoplasmic reticulum, smooth endoplasmic reticulum. As I explained to you in the previous slide, it is shown here. Cholesterol is esterified at the third carbon, as you see here, ester bond. A fatty acid is attached, done by acyl-CoA, that is fatty acyl-CoA, cholesterol, acyl transferase. And the type of fatty acid that is attached to the third carbon hydroxyl group of cholesterol, it is usually linoleic acid. Remember that. The major type of fatty acid that is esterified here is linoleic acid, and you know linoleic acid is an essential fatty acid. Now let's move on to see bile acid synthesis. Now the primary bile acids are synthesized in the liver from a cholesterol. That's one of the major fate of cholesterol that is made in the liver. Now a major type of bile acid that is synthesized in the liver is a cholic acid, which is found in largest amount in most mammals. And the other type of bile acid that is made in the liver is Chenodeoxycholic acid. That means a cholic acid and Chenodeoxycholic acid, these are the primary bile acids made from cholesterol, and that's how a cholesterol is used in the liver. Now the cholic acid and Chenodeoxycholic acid, further they can be conjugated in the hepatocytes into their glycine conjugation form or taurine conjugation form. We are going to see that in subsequent slides, and then they will be secreted into the biliary canaliculi, and then they will become part of the content in the gallbladder. When the gallbladder contracts, things will get into the duodenum, and in the duodenum they participate in emulsification process. Now when the primary bile acids reaches a duodenum, so in the duodenum, these primary bile acids, so they will be existing in their salt form, and that type of salt forms of bile acids are simply referred as bile salts. So I want you to know the names here like bile acids and bile salts, because we kind of use bile acids and bile salts synonymously when we refer to the actions of bile acids in the intestines. Now, very first enzyme which is involved in the synthesis of bile acids is 7-alpha hydroxylase enzyme. This 7-alpha hydroxylase enzyme, which initiates bile acid synthesis, is the most regulated enzyme in the bile acid synthesis. Let's get into the details how bile acids are synthesized in the hepatocytes. Now, cholesterol is diverted into bile acid synthesis by an enzyme 7-alpha hydroxylase. And this 7-alpha hydroxylase enzyme, it is a cytochrome P450 monooxygenase enzyme which belong to cytochrome P450 CYP7A1 subclass. So it is CYP7A1. So this is the type of cytochrome P450 that 7-alpha hydroxylase belongs to. Now the 7-alpha hydroxylase enzyme, as it is shown here in the figure, it needs vitamin C as a coenzyme, and it is a reductive biosynthesis here. That's why it needs NADPH plus H plus as the reductant, and also since it's a monooxygenase system of enzyme, so it needs a molecular oxygen here. So overall, cholesterol, it is hydroxylated at 7th carbon as it is shown in the figure to become 7-alpha hydroxycholesterol. Now this 7-alpha hydroxycholesterol can have two fates, which is catalyzed by 12-alpha hydroxylase enzyme. On one side, it will be converted into cholyl-CoA. On the other side, it can be converted into Chenodeoxycholic-CoA. Now in this process, the side chain of cholesterol, which is attached at 17th carbon, that 8 carbon hydrocarbon chain which is attached to the 17th carbon. So that will be broken down. 3 carbon moiety from that side chain will be released as propionyl-CoA. Remember this. So that means bile acid synthesis is one of the source of propionyl-CoA. And what happens to the propionyl-CoA? I have already explained you in my earlier lectures. Propionyl-CoA will be converted into methylmalonyl-CoA, and which is further converted into succinyl-CoA to enter into TCA cycle. Anyway, so cholyl-CoA is if you remove the CoA there, it is a cholic acid, and Chenodeoxycholic-CoA, if you remove a CoA which is there here, it becomes Chenodeoxycholic acid. Now the cholic acid and Chenodeoxycholic acid, they are the primary bile acids which are synthesized in the liver, and then they can be secreted into biliary canaliculi, but generally these are not the predominant form of secretion. The reason for that is cholic acid has got a pKa of 6. Similarly, Chenodeoxycholic acid has a pKa of 6. Since the cholic acid and Chenodeoxycholic acids, both of them, they have got pKa of 6, and if they are secreted in that form, if they reach intestine, so in the intestine also, the pH of intestine is around 6. That means we are working at pH equals to pKa. So whenever pH is equals to pKa, you know, the molecules exist 50% as protonated and 50% as deprotonated. Now the thing is, bile acids need to be deprotonated form. They need to be existing in predominantly deprotonated form. So that means their pKa should be lower in order to do that. With the pKa of 6 as cholic acid and Chenodeoxycholic acid, the bile acids are only 50% efficient in their detergent action in the intestine, because only 50% of the molecules will be protonated and 50% will be deprotonated, but only the deprotonated forms which are useful in the emulsification process, where the detergent action of bile acids are necessary for the digestion and absorption of dietary lipids. So how to improve that deprotonation process so that the efficiency of bile acids can be improved, so that their detergent action is available for emulsification and proper lipid digestion and absorption in the intestinal lumen. So the answer for that is conjugating these bile acids into their conjugated form. And the conjugation of bile acids will be going on in the peroxisomes. Remember that. Peroxisomes are the ones which will help in the conjugation of bile acids. And there are two kinds of conjugating molecules for bile acids. One is a taurine conjugation. That is, the taurine is a derivative of cysteine. Remember that. Taurine comes from cysteine. This cysteine is one of the fate of cysteine in our cells is making taurine. Now the taurine conjugation and glycine conjugation. That's what is shown here. Now similarly, Chenodeoxycholic acid also can undergo taurine conjugation and glycine conjugation. So once taurine is conjugated with cholic acid, it becomes taurocholic acid, and when the glycine is conjugated with cholic acid, it becomes glycocholic acid. Similarly, when taurine is conjugated with Chenodeoxycholic acid, so it becomes taurochenodeoxycholic acid. When glycine is conjugated with Chenodeoxycholic acid, so it becomes glycochenodeoxycholic acid. So with this conjugation, so what is the use of conjugation? So conjugation simply is going to decrease the pKa of cholic acid and Chenodeoxycholic acid. How? At taurine conjugation will decrease a pKa to 2. From 6, it will be decreased to 2. With the glycine conjugate will have pKa of 4. Overall, what happened here is from pKa 6, now it has decreased to pKa either 2 or 4. Okay, so what is the effect of this? So now the pH is 6 in the intestine. So pH, now compare pH and pKa. pH is 6. Now taurine conjugation is pKa2, so that is 2. So 6 and 2 now. 6 is more than 2. So pH is more than pKa. Whenever pH is more than pKa, the molecule will undergo deprotonation. So we are increasing the deprotonated form of these bile acids. Alright, with the glycine conjugation, so the pKa will be 4 and pH is 6 again, so there will be more of deprotonation. So the point here is, by decreasing the pKa, so a molecule is predominantly undergoing deprotonation whenever it gets into the intestine, and deprotonated form of bile acids, they are more efficient forms of bile acids because they will have higher detergent activity, and that's the point in a conjugation process. Now, out of taurine conjugate and glycine conjugate, so note that glycine conjugate is the predominant form of conjugated bile acids that we see in the gallbladder, which are going into the intestine for emulsification process. So moving on, so we have now a conjugated form of bile acids, which are also referred as a primary bile acids. So how many primary bile acids we have now after conjugation? So on the cholic acid side, we have taurocholic acid and glycocholic acid. So we have two there. And on the chenodeoxycholic acid side, we have taurochenodeoxycholic acid, glycochenodeoxycholic acid, two. So that means we have 4 primary bile acids. Now once they get into the duodenum, participate in emulsification process, digestion and absorption of lipids. So these bile acids, they move down towards the ileum, especially terminal ileum. As they move from duodenum towards the terminal ileum, so what will happen? The bacteria present in the intestine, so they are going to act on these primary bile acids. So they are going to do a deconjugation, means removal of taurine and glycine, and also they are going to cause 7-alpha dehydroxylation, and that's what is shown here in the figure. So after the deconjugation and 7-alpha dehydroxylation, cholic acid, that is glycocholic acid, taurocholic acid, they all will form deoxycholic acid. So that is what is a secondary bile acid after the action of bacteria. On the chenodeoxycholic acid side, they will become lithocholic acid, and that is the secondary bile acid. So we have 2 secondary bile acids coming from cholic acid side and chenodeoxycholic acid side, and that is deoxycholic acid and lithocholic acid. These two are relatively So these two are relatively less water soluble than the primary bile acids. Out of this, lithocholic acid is the one that is predominantly excreted out of the intestine in the feces. Otherwise, majority of these primary bile acids and the secondary bile acids from the duodenum going into terminal ileum, and in the terminal ileum, they are reabsorbed back. More than 95% of the bile acids which are reaching terminal ileum, they are reabsorbed back into the portal system, and they will be carried to the liver, and in the liver they will undergo a reconjugation process and they will get back into the duodenum again. So this is what is referred as enterohepatic circulation. So there will be around 98 to 99% of the bile acids will be reabsorbed and re-enter back. So only around 1 to 2% of the bile acid is lost, maximum of 5% I would say. So in the feces, and the loss of one bile acid in the feces is loss of cholesterol from our body. So out of the bile acid that is lost into the feces, it is predominantly the lithocholic acid, remember that. So here are some of the points related with bile acid synthesis. So we have glycine conjugates, which are glycocholic acid and glycochenodeoxycholic acid, which have got pKa approximately 4. And then we have taurine conjugates, which are taurocholic acid, taurochenodeoxycholic acid. They have a pKa of 2. And humans will have higher levels of glycine conjugate compared to taurine conjugate. So the glycine to taurine conjugate ratio is 3:1. Remember that point. So in the alkaline pH of bile, that is 7.6 to 8.4. The bile acids and their conjugates are assumed to be there in salt form. That's why they are called as bile salts. And the bile acids or bile salts are produced in the liver and secreted into the bile, and in the intestine the bacteria are going to deconjugate glycine and taurine and further dehydroxylase these molecules, and the action of intestinal bacteria will convert glyco or taurocholic acid into deoxycholic acid, which is a secondary bile acid, and glyco and Chenodeoxycholic acid will be converted into 𝐒𝐵𝐞𝐽 acid. That's a secondary bile acid. Now, greater than 95%, which is about like 98 to 99% of the bile acids are absorbed in the terminal ileum. So, the liver takes up both primary and secondary bile acids via enterohepatic circulation, as I explained to you earlier. They reconjugate them with glycine and taurine to make corresponding bile salts to release it into the biliary canaliculi, and that's how enterohepatic circulation of the bile salts is extremely efficient in reusing the bile salts. And as I said earlier, 𝐒𝐵𝐞𝐽 acid is the predominant form of secondary bile acid that is found in the feces. Only small fraction of, that is around 5% of the bile salts are escaping this absorption and therefore eliminated in the feces. Excretion of bile salts is the major route for removal of steroid nucleus, and that's the cholesterol removal from the body. So this is how cholesterol will be converted to bile acids, and bile acids will participate in emulsification, and in the process some of the bile acids are lost out of the intestine, and that's the loss of cholesterol from the body. Now the cholesterol, which is excreted from the body via bile acid either in the unesterified form or after the conversion of bile acid. So this is a way that excess cholesterol can also get out of our body into the feces, either as a free cholesterol or as a bile acid form. Now the predominant form of cholesterol that is found in the feces, it's a kind of converted form of cholesterol called as caprostenol, and this caprostenol is the principal sterol that is found in the feces, and it is the action of bacteria on the cholesterol, especially in the lower intestine like in the colon, to make cholesterol into caprostenol, and that is the excreted form of cholesterol in the feces. Remember that. Each day, pool of bile acids, which is around 3 to 5 gram, is cycled through intestine 6 to 10 times, and the amount of bile acid equivalent to the loss in the feces is synthesized from cholesterol, so that the pool of bile acid is constantly maintained. So this is how bile acids are synthesized in the liver, and that's how bile acid levels are maintained. Coming to the regulation of bile acid synthesis, so as I said in the beginning of this chapter, 7-alpha hydroxylase enzyme is the most regulated enzyme, and also it is catalyzing the rate limiting step for bile acid synthesis. And this 7-alpha hydroxylase enzyme, I told you, it belongs to cytochrome P450 class of enzyme, CYP7A1. And the activity of this 7-alpha hydroxylase enzyme is feedback regulated by a nuclear bile acid binding receptor, which is called as FXR receptor, that is farnesoid X receptor. When the size of bile acid pool in the enterohepatic circulation increases, now the FXR receptor is activated, and the transcription of CYP7A1 gene will be suppressed. This is a kind of a negative feedback effect in response to higher levels of bile acids in the enterohepatic circulation. Out of two primary bile acids, that is cholic acid and Chenodeoxycholic acid, Chenodeoxycholic acid is of particular importance in activating FXR receptor and thereby regulating the bile acid levels in the body. Remember that point. Now CYP7A1 activity is also enhanced by cholesterol of endogenous and dietary origin, and also it is regulated by hormones like insulin, glucagon, glucocorticoids and thyroid hormones. So that's how bile acids are synthesized from the cholesterol by variety of steps into all the way into cholic acid and chenodeoxycholic acid, and the important enzyme there is 7-alpha-hydroxylase. So they are conjugated in the peroxisomes so that bile acid efficiency in the intestine increases in the emulsification process. And when the bile acids are moving to the terminal ileum, so bacteria will act on bile acids and convert the primary bile acids into secondary bile acids. And primary bile acids and secondary bile acids, they will be reabsorbed and get into the liver by enterohepatic circulation to come back into the intestine after reconjugation process. So, and that's how the efficient enterohepatic recirculation will maintain a bile acid pool. Only around 5% of the bile acid is lost in the feces, and within that lithocolic acid is the predominant one. And the excess cholesterol which is lost out of the liver entering into the biliary secretion going into the intestine, so it can be converted into captostenol, and that is done by the bacteria in the colon, and that's how it gets out of the body. And the bile acids are regulated at 7-alpha-hydroxylase enzyme step, and the regulation is mediated by a nuclear receptor called farnesoid X receptor. So that's about bile acid synthesis. I hope this video has helped you in understanding this very important concept. Thank you.